Good morning everyone. Today I will teach you the next topic, that is the experimental proof of the replication, such as, or you can say, that is the Meselson and Stahl experiment. As you know about the replication, what do you mean by replication? The replication is the process of the synthesis of DNA. And during the process of replication, the DNA is duplicated. The DNA is going to be increased in their numbers. During the process of the cell division, the process of replication, or you can say the synthesis of DNA, takes place in the S phase, that is synthetic phase of the cell cycle. The DNA replication takes place by semi-conservative means. So before the experimental proof of the semi-conservative method of the DNA replication, first we will talk about what is semi-conservative mode of replication. In the semi-conservative mode of the replication, the double-stranded DNA, when it is going to be replicated, or when the double-stranded DNA is going to synthesize the new strand, the newly synthesized strand has one parent DNA, and while the one strand is newly synthesized. You can see here, this is the parent DNA. The parent DNA have how many strands? Each DNA molecule, as you know, is made up of how many strands? 2 strands. And at the time of, suppose these are the strands of the parent DNA. At the time of DNA replication, both the strands, they are separated to each other, both the parent strand. And each strand acts as the template strand. You can say this one is the template. This also acts as the template strand. Means the double-stranded DNA. At the time of replication, both the strands, the hydrogen bonds are broken down, and both the strands of the DNA duplex, or you can say the DNA, are separated to each other. Both the strands, they are separated to each other, and these strands act as the template. Now, as per the template strand, the new strand is synthesized, and when the duplex DNA is completely replicated, the newly synthesized DNA has one parental strand and one newly synthesized. This one will be also the parental, and this will be the newly synthesized strand. So in semi-conservative mode of replication, it's very, very important. In semi-conservative mode of replication, the newly synthesized DNA strand has one parental strand and one strand is newly synthesized. So out of 2 strands, one is old while another one is the new. You can see in this diagram I have drawn here with this green sketch. This is the parent strand, a DNA molecule. Now both the strands act as the template, and now according to the old strand, the new strand is going to be synthesized here also. And from one DNA molecule, when it undergoes into the process of replication, one duplex DNA synthesizes into 2 DNA molecules, and in each DNA molecule, one strand is old while another one is newly synthesized. As you know, during the process of replication, I will teach you so many enzymes, so many proteins, they are required, and as per base complementary rules, because one strand is just complementary to the another strand, and as per base complementary rules, the new strand is synthesized from the template one. But today we are not going to discuss about the steps of the DNA replication, that how the process of synthesis of the DNA takes place. We will not study today. We will discuss in the next video that how the process of replication takes place. But the topic which we will discuss today, that is experimental proof of the DNA replication, which is semi-conservative method. These two scientists, Matthew Meselson, simply you can say Meselson, and Franklin Stahl, these two scientists, they experimentally proved the semi-conservative method of DNA replication. So let us start that how they did their experiment, what were the steps, and how they proved that the method of the DNA replication is semi-conservative. You can see here, I have drawn here so many diagrams. In the semi-conservative mode of DNA replication, first of all, you can see here, this is the double-stranded, or you can say duplex DNA. Two strands are there. As you know, in each DNA molecule is made up of how many polynucleotide chains? 2 polynucleotide chains. In the DNA replication, to prove the semi-conservative method of the DNA replication, first of all, these two scientists, they prepared a culture medium and they added 15 NH4Cl. They used 15 NH4Cl ammonium chloride. As you know, 15N means the 15 nitrogen. The 15 nitrogen, the source was ammonium chloride. The full form, the 15 NH4Cl means NH4Cl is the formula of ammonium chloride, and he used the 15 nitrogen. 15 nitrogen is the isotope of the nitrogen. Actually, there are two isotopes of the nitrogen, and these are 15N and 14N. The 15N isotope is the heavy isotope and 14N is the lighter isotope. In the natural condition, the 14 nitrogen, 14N isotope is found. But in this experiment, first of all, he added the heavy isotope, that is 15N, into the culture medium. And inside the culture medium, he transferred E. coli bacteria. As you know, E. coli bacteria, that is Escherichia coli bacteria, is a very common bacteria, and this bacteria has been used to demonstrate so many experiments related to the molecular genetics. And in this experiment also, the experimental organism was E. coli bacteria. So, first he added 15 nitrogen, 15N isotope into the culture medium, and then he transferred E. coli bacteria, their culture, and he observed that the DNA molecules of E. coli bacteria were retained by 15N, or you can say the 15N heavy isotopes. The 15N is not the radioactive substance. You have to keep in your mind 15N, 15 nitrogen is the heavy isotope of the nitrogen. And when he used 15 nitrogen isotope, the nitrogen is already present in the nucleotide of the DNA. As you know, nitrogen bases are to be there, and all these nitrogen bases, the nitrogen, were retained by 15N isotope. So this one is the, you can see here, I have drawn this diagram with the green sketch pen, that it indicates that the DNA molecule of the E. coli has been retained by 15N nitrogen. In this experiment, E. coli bacteria were used to demonstrate this semi-conservative method of the replication. Actually, E. coli bacteria can multiply very rapidly, very fast, you can say. And in bacteria, within 20 minutes, the bacteria can multiply, can produce the new bacteria, or you can say the DNA is also going to be replicated, synthesized just within 20 minutes. So after 20 minutes, it can produce a number of E. coli bacteria. So the multiplication is very fast, and this was also a reason to select E. coli bacteria to demonstrate this experiment. So first he used in the culture medium, that is the isotope of the nitrogen, and that was the heavy isotope 15N, and he cultured the E. coli bacteria for several generations, and all the E. coli bacteria, their DNA molecules were retained by 15N heavy isotope. Then when all the bacteria, their the DNA molecules, they were retained by the 15N heavy isotope, then what he did in the second step, then he transferred this bacteria having 15N into a culture media, into a medium containing 14N ammonium chloride, or you can say the 14, and that was the lighter isotope of the nitrogen, or you can say the normal isotope of the nitrogen. And after 20 minutes, when he added the 15N E. coli bacteria into the fresh medium containing 14N, because after 20 minutes they have been multiplied. After 20 minutes, what he observed? He observed the hybrid. This is the hybrid DNA. Hybrid DNA means during the process of, as you know, replication, both the template, both the strands, they are going to be separated to each other, and now both the strands, they act as the template to synthesize the new strand, and all the new strands which are synthesized after 20 minutes, means during the process of DNA replication, all the newly synthesized strand, now they have retained by which nitrogen? 14N. Why? Because 14N was added into the fresh medium, into the culture medium. So here 50% result he observed. And how he observed? He observed by centrifugation. In the centrifugation, he used cesium chloride density gradient by cesium chloride density, because cesium chloride is used for the centrifugation. As you know, in the centrifugation inside the test tube, when they centrifuged the DNA molecules, the heavy DNA molecules, they are settled down on the bottom, and the lighter one, if the DNA molecule will be lighter one, it will be deposited just above to the heavier one. So in the centrifugation, the DNA molecules which are heavier, containing 15N, will settle down, will be settled on the bottom, and the lighter one, which are going to be retained by 14N, will be settled down just above to the heavier ones. So two different layers are formed. The heavier ones are down and the lighter one will be on the upper side. So he observed, you can see here in this test tube, because he added the heavier one only, this 15N nitrogen, that's why only one zone he observed after the centrifugation, and all this DNA containing what kind of the isotope, 15N. But after 20 minutes, when he added 14N into the culture medium, then he observed the 50% was containing 50% DNA are containing 15N, while 50% which are newly synthesized, now they are going to be retained by 14N isotope. And when he observed in the centrifugation the two different layers, he found the 15N, because it is 15N, the DNA molecule, the 15N are the heavier, settle down on the bottom, and 14N just above, because 14N DNA are the DNA molecules which are labeled by 14N or which retain 14N, they are deposited just above. Overall, in the first generation, he obtained hybrid DNA. Hybrid means 50% DNA that had the 15N nitrogen and 50% that had the 14N isotope of the nitrogen. Again after 20 minutes, if you will consider from here, again after 20 minutes, means after 40 minutes, if you will consider the timing from the first one, from the parent one, it will be the 40 minutes. And if you will consider from the generation first to generation second, it will be the 20 minutes only. So I have written here 40 minutes in your NCERT book because the time has been considered from here. So after 40 minutes, again the 50% DNA he obtained hybrid, and remaining 50% he obtained the lighter ones. You can see here, this one is the old strand containing 15N, this is for the 15N, this is for 14N, again this one is for the 14N, and this one is the 15N. So overall, the 50% DNA molecules which are synthesized in the second generation are hybrid. This one is the hybrid, and remaining 50% they are lighter, containing what kind of the isotope? 14N. And he observed here also in the centrifugation, in the cesium chloride density gradient, 50% was completely, the 50% DNA was completely having the 14N isotope of the nitrogen, and remaining 50% had the hybrid DNA containing 14N and 15N both. So gradually, the amount of this parental DNA which was added, that was 15N, their percentage will be decreased. And when he studied the amount of 15 and 14N into the next medium containing 14N solution, he obtained that the 14N amount gradually it was increased, and overall this experiment proves that the method of DNA replication is what? Semi-conservative. So overall, Meselson and Stahl experiment proved that the DNA replication is semi-conservative. So once again, you can keep in your mind the topic which we discussed today. As you know, the DNA replication is the process of the synthesis of DNA, and the process of replication is semi-conservative type. That semi-conservative means when the new strand of the DNA molecule is synthesized out of two strands, one is retained from the parent or one is old strand, while another one is the newly synthesized strand. And if out of two strands one is old and one is new, such type of DNA replication is called semi-conservative method of DNA replication. And that semi-conservative method of the DNA replication was experimentally proved that the method is semi-conservative by these two scientists, Meselson and Stahl. In this experiment, they used the two different isotopes of the nitrogen, which are 15N and 14N. In the first step, they used 15N into the culture media, and in the culture media, then they added E. coli bacteria, because E. coli bacteria can multiply very fast. Just within 20 minutes, it can produce the number of offspring. Within 20 minutes, the DNA is also going to be replicated. When he added 15N isotope, 15N isotope of the nitrogen, then all the parental DNA and all even the newly synthesized DNA also in that particular culture medium in which he used the 15N, all the bacteria, their DNA molecules were retained by 15N isotope. Then what he did in the second step, he added 14N isotope of the nitrogen into the different culture media, and he transferred the E. coli bacteria having 15N into the 14N into the fresh medium, and he observed into the first generation that all the bacteria, their DNA molecule, 50% they had the 15N isotope, they were incorporated, and remaining 50% they had the 14N isotope. Again, after 20 minutes, he observed that the 50% they were hybrid and remaining 50% they were lighter. And at the different intervals, he took the samples and studied by centrifugation by using cesium chloride density gradient. And finally, he proved the method of DNA replication is semi-conservative. So that's all for today. Tomorrow we will discuss about the DNA replication. We will also study about that how many enzymes, how many proteins they are involved for the process of DNA replication. So that's all for today. Thanks everyone.